All right, excellent. So let's go ahead and get started. Today is Friday, June 11th, the end of our first week, although really 2 weeks worth of material because we're just rapidly going through it. Welcome to summer school. Today we continue our discussion of the cell. We talked about the nucleus, we talked about the cytoplasm, so now we have to talk about the plasma membrane, what it's made up of, what it does, and how it works. And then, like I said, for lab today, we are going to do our practice lab exam. So that you'll have that opportunity to see questions that are identical or similar to questions that are going to be on your exam, and we can talk about it. And it's a good gauge to see how you're studying so far as we rapidly approach the first exam. Next week is a busy week. We have our Unit 4 review and our Labster membrane transport due. Our first Physio X, which remember has 5 activities, so that's 5 different lab reports. 2 more unit reviews, 5 and 6, and then a couple other fun activities. There's a biointeractive handout on skin color. That's a really interesting activity you're going to do. There's a fun fingerprint activity. The fingerprint activity in your lab manual is a fun activity, but obviously it's not something we can do where basically one of you is the bandit and you have to figure out by looking at everybody's fingerprints who the bad guy is or anything like that. But I do want you to have the opportunity to either for yourself or the people in your family, take a fingerprint of yourself and using the minutia details, the Galton points and the characteristics that are listed on page 146, identify some of the characteristics of your fingerprint and be able to do that activity. So that's kind of fun. The other major thing that's due Friday next week, and we'll obviously talk about it next week more when we get to the tissues, is on Canvas, there is an epithelial tissue and a connective tissue handout. These handouts, as I talked about on the previous class, and we will talk about more next week when we get to them, these are 2 things. They are obviously an assignment, but it is also your study guide for that portion of your lab and lecture exam. My guess is easily a third to a half of your exam is going to involve epithelial tissues. Probably about a third of your exam is going to be an assessment of your histology, your epithelial tissue and connective tissues. And what those handouts are is a list of the tissues that you will be responsible for on histology slides, and the materials on each of those slides that you are responsible for as well, with, in parentheses, the key term I will use. We will try to get through as much of that together as we can next week, but it's probably a good thing to start looking ahead at it. And to emphasize to you how important histology is going to be in this class, you are going to be responsible for drawing those tissues, labeling those tissues, and turning them into me. Now, obviously, you've seen my drawing skills on the whiteboard. I'm not grading anybody on their drawing skills. However, what I want this to be is an effective study guide for you. So you're going to draw it however it makes sense to you, label the things that are on there, and you will be scanning those or taking pictures of those and turning those into me. And so not only will you get the points for doing it, but then it is also going to be an excellent study guide for you over the weekend going into the lab and lecture exam as well. Like I said, we will do at least some of it together, if not all of it together. We will get to that next week. But again, we've only got 2 weekends before the exam. At the fast pace, it's not a horrible idea to start looking ahead at some of these things. All right. Again, Monday the 21st, lab and lecture exam. That is all we're doing on those days. So you'll have the entire class time to take those 2 exams. And like I said, we'll talk about that more when we do our practice exam today. All right. Questions on the game plan for the next week and change? All right, excellent. Then let's dive into, back into ourselves really, and talk about our plasma membrane. Now, what is the function, or really we should say functions, plural, of our plasma membrane? What does our plasma membrane do for the cell?
Keep certain things in and certain things out.
Excellent, I like that. It forms what we call a semi-permeable boundary, right? Where it allows some things to get into and out of the cell, but it limits the movement of other things. All right, it's like the walls of your house. The walls of your house basically form the boundary of your house, but things are able to get in and out. Some of those are the things that you want to get in and out, like your loved ones, right? Some of them are things that you may not necessarily want to be able to get inside and out, like your mother-in-law or your bugs and insects and other things along those lines. And there are limits to where things can get in, windows, doors, things along those lines as well, right? So we're able to be semi-permeable, allow some things in and out, but still form that important boundary. And as we talked about, what is the primary component, the primary macromolecule that forms our plasma membrane?
Phospholipids?
Phospholipids, absolutely. And if you remember correctly, a phospholipid is key in that it has a polar head to it, and then a fatty acid tail region to it. And how are these phospholipids arranged?
In a
Double
bilayer.
bilayer.
In a bilayer, absolutely. So we have tails in, heads out. We have this arrangement of the phospholipids, and I'll draw 3 or 4 of them here. So there's a way to just cut and paste these things. Excellent. So we have our phospholipid bilayer, which we know is the basis of our plasma membrane. But there are a lot of other components to this as well, and some important reasons from it. Now let's start first, one more thing about these phospholipids. All right, phospholipids, as we know, the tail part is hydrophobic, the head part is hydrophilic, and that means that the tails don't like water, whereas the heads do. Now, if I had a glass of water and I poured a little bit of vegetable oil really, really carefully onto the surface of that glass of water, what would happen?
Stay separated, the oil will float.
Right, the oil would float. It would form a pool of oil on top of the water, separate from the water. And if we were to look at it at a microscopic level, does each individual molecule of the fat just find a location where it's happy with and just sit there? I always imagine in my mind, I think back to when my eldest was in kindergarten, right? And we would go to the kindergarten because they'd be putting on some kind of show or presentation or singing some song or had to do their exercise. And the first thing that the teacher would do is tell them to spread out. So what did all the kids do? They all stood up and they swung their arms around, and they kept moving till they got a point where when they swung their arms, they didn't touch anybody else. So this was my space with my arms and somebody be on the other space. And is that what our fat molecules do? They just find their location, swing their arms around till they're not bumping into anybody else, and they just stay right there? Is that what happens? No, these things are constantly moving. We actually have a name for that. The name for this is it is a fluid mosaic. What that means, and again, these numbers aren't real, but I'm using them for our argument's sake. Let's say that every second, one of these phospholipids changes position with another phospholipid. So these are constantly moving around. However, they're just moving within their layer, right? One of these phospholipids can't change from one layer to another, can they?
No.
Well, I'm asking the question, what should the obvious answer be?
No.
Well, if it was no, then I wouldn't be asking the question, right? Because then I would be obvious.
Yes.
But
Yes,
I'm
it
asking the
can.
question, so what's the obvious answer?
Yes.
Yes. Now, is it going to be as easy to switch layers as it is to switch position in a layer?
No.
No. If one of these is changing every second, then they're changing layers like once a day, right? Again, those numbers aren't the real numbers, but it gives you an idea of the speed of these types of things. So these things are moving around, and that's a good thing. That flexibility of our plasma membrane, that looseness of our plasma membrane serves some important functions. However, it could also be too fluid. If it's too fluid, then what's going to happen is that it's not going to form that barrier that we need. So one of the ways we can limit the movement of this is by using another lipid, your friend and mine, cholesterol. Remember, we talked about how cholesterol everybody thinks is a bad thing, Remember, cholesterol is made up of those 4 carbon rings and a little bit of a functional group on it. But what'll happen is this cholesterol will line up adjacent to those fatty acids. And now if you think about it, if we were to name these 3 phospholipids, A and B and C, which ones are going to be more likely to change positions, B and C or A and B, with that cholesterol in the way?
B and
B and C.
B and C. So cholesterol in this case, this type of cholesterol is actually a good cholesterol. It helps to actually give a little bit more structure and a little bit more integrity to our phospholipid bilayer. So again, one of the important structures, one of the primary structures of our plasma membrane are those membrane lipids, phospholipids, but also that cholesterol as well. However, lipids are not the only thing that we find in our plasma membrane. There are also going to be some carbohydrates. There are several carbohydrates, different types of carbohydrates. Most of these carbohydrates are found on the outer surface. So let's go ahead and let's erase a couple of these arrows. Many of our carbohydrates are found more commonly on the outer surface. So we'll have a row of glucoses on the outer surface of this. And what might the function of this glucose on the outer surface be? So energy is definitely one of the main functions of sugar, and having some of that sugar on the outer surface absolutely could be useful, and some of it could be there to provide energy close by for any kind of metabolic processes that need to take place. But I'll give you another hint. These glycocalyces that are on the, and again, that's not wrong. That is true. There can be some used for energy. But let's think of the other function. This glycocalyx is also known as a sugar coat. Right? And what happens to your hand when it gets coated in sugar?
it becomes more of a rough surface.
It becomes a rough surface, and what else?
Sticky.
Sticky, absolutely. Helping it to attach to the surrounding tissue, helping it to be able to anchor to other cells. So that sugar coat can help it to stay in place within the tissue. So we have this glycocalyx, this sugar coat on the outer surface, which can help to anchor it, can provide energy, and it can serve one other function as well. All right. Again, today is Friday. The weekend is coming up. So I'm sure, like me, you guys are planning on going to Vegas for the weekend. Because once again, I'm going to try to win some money so I can take the rest of the summer off. I can quit this job, don't have to take this class, and we can just, live in the lap of luxury. So, of course, it's a last-minute flight, so I've got my black rolling bag, and by the time I get there, I can't bring it onto the plane anymore, and I have to check it. So I get to Vegas, I get to the carousel, a luggage carousel, and how many black rolling bags come out of that luggage carousel?
Lot.
A lot. So how do I know which one is mine?
Your name tags on it.
Yeah,
some sort of
absolutely.
marker.
And again, I've got some kind of marks on the outer surface. Yeah, it's got the name tag, but those name tags are tiny. So I need something that really helps me to be able to distinguish mine. So mine has a nice, big, huge green bow on it and a big One Direction sticker, right? And so when I see that One Direction sticker, I know that one's mine and I can grab it. So we need some type of identifier on the outer surface. Is it important for us to know which cells are us and which cells are not us?
Yes.
Yeah, absolutely. So having these types of tags on the outer surface can be a very important function of that plasma membrane. Excellent. So we have those lipids, we have those carbohydrates, and if we're going to have lipids and we're going to have carbohydrates, not surprisingly, we are going to have membrane proteins as well. Now, membrane proteins come in 2 flavors, integral and peripheral. What do you think the difference between these two are? What do you think the difference between an integral membrane protein and a peripheral membrane protein is?
One is inside, one is on the out.
Not a bad guess. So inside and out, but let's be more specific. Inside or out of what? The cell?
The membrane. Like
Exactly. That's a great way of describing it. An integral membrane protein is a membrane protein that is embedded in one or both layers of the plasma membrane. Whereas a peripheral membrane protein is a protein that is associated with the plasma membrane, but not connected to one of the layers. Let's go ahead and draw these on our ever expanding phospholipid bilayers. So one example of an integral membrane protein could be something like this that goes through both layers of the plasma membrane. In this case, this would be an integral membrane protein. But because it goes through both layers, we would actually also call this a transmembrane protein. Because it goes all the way through. Others may just be embedded within one layer. This is still an integral membrane protein because it is embedded within one part of the phospholipid bilayer. But obviously this one's not going through both layers, so it is not a transmembrane protein. Another protein, however, may be here, right? It could actually be attached to an integral membrane protein, but it is not actually attached to the plasma membrane itself. And because it's not actually attached to the plasma membrane itself, this would be a peripheral membrane protein. So integral membrane proteins are attached to the plasma membrane, embedded in one or both layers. A peripheral membrane protein is associated with the plasma membrane, but doesn't actually connect to it. And an integral membrane protein that goes through both layers, we call a transmembrane protein. Now, we know that proteins are the most diverse in their structure, and so they're also the most diverse in their function. So what are some of the possible functions that these proteins could possibly have for us in a plasma membrane?
To help transport between.
Yeah. Absolutely. That transmembrane protein could allow substances to come into and out of the cell. It could also be a receptor for some type of chemical signal, like a hormone or some other type of chemical signal to affect some type of change. Enzymes to do some function. These can also be tags on the outer surface and all sorts of other fun things that they can do as well. So notice when we're drawing that cell and we just draw a big circle on the outside, that line that is a circle has a whole heck of a lot that's going on. And while I've done a truly amazing job of drawing this, let's take a look at the pretty picture from your textbook that shows this as well. Here we can see all of those components that we were just talking about. Again, the phospholipid bilayer is the primary structure of our plasma membrane. Notice we have some of those cholesterols embedded within the fatty acid tails, providing a little bit more structure, a little bit more integrity. Notice that the majority of the glycocalyx, the sugars, are located on the outer surface, providing those tags, providing that attachment, providing that energy. Some proteins are integral membrane proteins embedded in one or both layers. Notice this one here has a hole through it, so clearly it's allowing something to come in or out. Notice there's plenty of peripheral membrane proteins. Most of them are on the inside, but notice there can be a peripheral membrane protein on the outside as well. These can be things like receptors for chemical signals, enzymes, all sorts of other things, or tags, again, that help us to identify this material. So we can see that line has a whole lot of stuff going on with it. Now, I want to get to, as I mentioned, there are attachments to these proteins, markers, enzymes, receptors, all sorts of things. But as I also mentioned, these proteins can either be channels or they can be transporters. They can form pores or they can form movers. And what do you think the big difference between a channel and a transporter is? What's the difference between a channel and a transporter?
Transporter requires energy, ATP.
Excellent. Transporter requires ATP. Any idea what we call it when this kind of movement requires ATP?
Active transport.
Yeah, so this is going to use active transport, whereas a channel does not require ATP. So what do we call that?
Passive.
Passive. Excellent. So not only do we need to talk about the anatomy and function of the plasma membrane, but we also need to talk about these important concepts of active and passive transport. All right, questions on this? Excellent. So like I said, we've got this pretty picture now of our plasma membrane, and we need to talk about the role that it plays in the helping of the movement of our materials. All right. However, we don't have to limit ourselves to a plasma membrane yet to really understand these concepts of active and passive movement, right? Instead of a cell, we can go ahead and erase this amazing looking thing. And instead, we can have a big, huge pitcher. And that big, huge pitcher can be filled with water. And we can take a packet of Kool-Aid. And of course, if we're going to do Kool-Aid, oops, hold on. It has to be grape because grape is the best. And we are going to pour that Kool-Aid into my pitcher of water. Now, when I pour that water, when I pour that Kool-Aid into that water, do the crystals of Kool-Aid just stay clumped together, shivering and hiding in the corner of the pitcher?
No.
No, they diffuse, absolutely. And does that diffusion require ATP?
No.
No, diffusion is a passive process. It basically just uses the kinetic energy of the molecule. And what's the key to the movement of that kinetic energy for that molecule?
High concentration to low concentration.
True, but you've got the right idea. But in fact, the movement is random. It's not a directed movement. But when we have a disparity like this, you are absolutely correct. The key to diffusion is we get a net movement from a high concentration to the low concentration. So with diffusion, we get this net movement where this Kool-Aid is going to spread out inside of this beaker until it reaches a point where it's all pretty much equally spread out from each other. What do we call that magical point where it's all equally spread out from each other?
Homeostasis.
True, homeostasis is one term for it, and that is accurate. However, when we're talking about the movement of substances in a solute, we have another term for that. There we go. Veng's got it. Equilibrium. Oops. Equilibrium is this point where it is uniformly diffused around. Now, again, are these Kool-Aid molecules like those kindergartners just finding their spot and staying in that one location?
Of course not.
No. The key to equilibrium isn't that there's no longer movement. Remember, there is movement. It is random. But the key to equilibrium is that there is no net change. So what happens is for every Kool-Aid molecule that moves to the right, one moves to the left. For every one that moves down, one moves up. Right? You could sit and stare at a pitcher of Kool-Aid from now to the end of time. While you're sitting there staring at that pitcher of Kool-Aid, is there ever going to be a period of time where suddenly all of the Kool-Aid moves out of one region of that pitcher and that portion of the pitcher is now clear?
No.
No. Once we reach this equilibrium, things are still moving, but the key to this point is that there is no net change. All right? And that is our goal. Molecules want to move on their own passively without ATP to reach equilibrium. And to do that, they move from a high concentration to a low concentration. Now, that doesn't mean that all diffusion is equal. We can change the rate of diffusion. Oops, didn't need that to be all capital S. What kind of things can I do, or what kind of factors will change the rate of diffusion?
Heat,
Temperature.
mechanical.
Okay, yes, you're right. If I'm making Kool-Aid, one of the things I'm going to do is I'm not going to sit there and passively wait for it to reach equilibrium. I'm going to stick my spoon in there and move it around. However, if I'm doing that, I'm adding energy. And if I'm adding energy, then it's no longer diffusion. So what kind of, so if I'm just pouring a crystal into the water and letting it reach equilibrium on its own, what are some of the factors that can make it go faster or slower?
Temperature.
Temperature, absolutely. With temperature, if I increase temperature, what happens to the rate of diffusion?
Speeds it up.
It goes faster as well.
Faster.
Excellent. What else? What else can affect the rate of diffusion?
pH.
It can, but not necessarily. It will for some things, but not for others. You are right. We want more general universal rules here, though. Okay, I
Size.
like farther apart. I like that. Distance, right? Let's think about this. If I had two pitchers, each contained 1 liter of Kool-Aid, and one was big and tall, and one was short and fat, and I put the same amount of crystals in them, would they reach equilibrium at the same point?
No.
Exactly. The one that it has to travel further in is actually going to take longer to reach equilibrium. So absolutely, the distance things have to travel, and I'm going to go ahead and erase these so that I can get it out of the way so I can do more writing. But you're absolutely correct. The distance that it has to travel definitely affects diffusion. The shorter the distance, the faster it's going to reach equilibrium. What else?
Volume.
Well, I like both of those things. Okay, so concentration gradient and solvent density kind of are the exact same thing. So I like that. Let's look at another example. I have 2 pitchers of water. And this time, just because I want to increase the rate or affect the rate of diffusion, I'm going to put a big sheet of saran wrap down the center of it. On this side, I'm going to put 75% Kool-Aid. And on this side, I'm going to put 50% Kool-Aid. And this is beaker A. And this is beaker B. I then take a screwdriver, and with that screwdriver, I poke 3 holes in that saran wrap. Now, if I do this, is the rate of diffusion from left to right going to be the same in both of these beakers?
No.
No. Which one's going to be faster, A or B?
B.
All right, raise your hand if you say A. Or hit the raise your hand button, hit the yes button. Raise your hand or say yes if you say B. All right, raise your hand if you didn't raise your hand. Excellent. Why didn't you raise your hand? Because you didn't know the answer, or because you knew I didn't give you enough information, right? Notice we only know what's going on one side of this beaker. What if I told you on this side of A, it was 70% Kool-Aid, and on this side of B, it was 10% Kool-Aid? Which one moves faster now, A or B?
B.
B, right? Notice, as someone mentioned, it's not just the solvent density, but it is the concentration gradient, right? Here, the difference is only 5. It's a relatively small difference, and so it's going to be a relatively low rate of diffusion. Here, the rate of diffusion is going to be much faster because there is a bigger difference. So absolutely, concentration gradient makes a difference. Notice also, if I had 2 identical environments, right? So I had 75 on this side and 70 on this side, just like in A. But remember, on the top one, I said I used my screwdriver and I poked 3 holes. But what if on the bottom one, I use my screwdriver and I poke 6 holes? Are both going to diffuse at the same rate?
No.
No.
No. Which one does more, does faster?
B.
B, right? This is an example of that term we used before, surface area. By putting more holes, we have more surface area for the material to be able to pass through, right? So more exchange can take place. Excellent. I can think of at least one more factor, that can influence and change rate of diffusion. Anybody know what else, what other
Pressure?
factor? True, although pressure can, but often pressure is something that is hard for us to be able to modify, and it kind of affects things early. Pressure is very important for gases. So there are some things that pressure is going to be very, very important for. Pressure is really not going to affect Kool-Aid. So when we're pouring the Kool-Aid, the crystals in the water, it's not going to have
Volume.
much effect. But what do you mean by volume?
Volume of the container, like size of the container.
True, absolutely. And I think that that definitely would be a case, although you could argue that would influence the distance that the molecule has to travel. So that might be the same thing as this. There you go. That's the other one I was thinking of. The mass of the molecule, or the size. Right? What diffuses faster, big molecules or little molecules?
Little.
Little molecules, absolutely. So excellent. So those are 5. Let's see what I came up with on my list from the lecture. Oh, hold on. We'll get to that in a second. Here we go. All right. Concentration gradient, temperature, size, distance, surface area. Excellent. We've hit it all. So those were the big things. Emily, did you have a question, or is your hand still up from before? Okay, perfect.
Can't come down.
Excellent. Excellent, excellent. So when we talk about diffusion, these are the factors that can influence it. All right. Now, as we mentioned, our plasma membrane is semi-permeable. It will allow some things to just pass right on through it. Oh, I erased my previous picture. All right, so we'll cheat and go back. So let's draw the simplified version of the plasma membrane. There it is. We mentioned how that plasma membrane is semi-permeable. Some things are able to just pass right on through the plasma membrane without any help at all. We have our phospholipids, we have their fatty acid tails, but some things can sneak right on through without any help at all, right? What kind of things are able to pass through our plasma membrane without any assistance at all?
Water.
Water is a tricky one. We'll put water on the list, but we have to come back to water. What else?
Lipid-based.
Excellent. Likes like. So something that is lipid-based will definitely pass through the plasma membrane. What else? If I have a lipid-based molecule the size of a 18-wheeler, is it going to easily pass through the plasma membrane?
No.
No, it's got to
No.
be small.
Got to be small things, right? And what's so special about lipid-based materials that allow it to pass through?
They're non-polar.
Yeah. They are non-polar, or they're hydrophobic. Excellent. So things that are non-polar or hydrophobic, things that are small, or things that are lipid-based are all things that can easily pass through the phospholipid bilayer without any assistance at all. We have a special name for that. We just call it simple diffusion, right? Pass through the plasma membrane without any help. Lipid-based, small, non-polar. And that brings us back to water. Does water meet those three criteria?
No.
No, it is small, but it is definitely not lipid-based, and it's definitely not non-polar. So when I asked what passes through the plasma membrane without any help at all, the first thing you guys mentioned was water. Should water be able to pass through the plasma membrane without any help? No. But you all said it first because you all know it's true. Water does. And what's interesting is we don't fully understand how. They have some theories, and the current theory goes along this line. As we know, these phospholipids are a fluid mosaic. And as we mentioned, they are constantly moving. So what they believe happens is that when the phospholipids are changing positions, as they're shifting in relationship to each other, they form small gaps. And those small gaps are enough for water to basically sneak through the cracks and get into and out of the plasma membrane. Water is so ubiquitous, there's so much of it and such a strong driving force on it that with those little movements of the phospholipid, water is able to take advantage of those small gaps, those small openings, and sneak into and out of the cell. So yes, water does indeed move via simple diffusion. Of course, we also know that anatomists hate us. So while it is 100% accurate to say that water moves via simple diffusion, they've actually given a name to the simple diffusion of water. And what is that fancy name they've given to the simple diffusion of water?
Osmosis.
There you go. Osmosis isn't some mystical, magical characteristic of water. It is just merely simple diffusion of water. It's a fancy name for the simple diffusion of water. So again, things that are small, things that are non-polar, things that are lipid-based can get in via simple diffusion. Water shouldn't be able to, but it does, called simple diffusion, or as we mentioned for water, that's also osmosis. Anything else that wants to get in needs some type of protein to help it. So anything else, our ions or other larger molecules or things like that, need help of a protein. And since it needs the help of a protein, we call this facilitated. Facilitated because it needs the help of a protein. Remember, channels are passive, so we can have a facilitated diffusion. Oops. Or transporters, remember, use ATP, so that would just be facilitated transport. All right, so again, this one is passive. This one is active. Both need proteins, so the difference is whether or not we need to use ATP or not. And we'll actually see these membrane transports in transport mechanisms in just a minute. However, things that are really, really big, we need another way to get them into and out of the cell. And we can do that via vesicles. We've already talked about one example of this because we know in our Golgi apparatus, we can bundle a bunch of proteins into a vesicle, bring it to the plasma membrane, attach it to the plasma membrane, and release those contents. And what did we call that process?
So cytosis.
Exocytosis. Excellent. That's our way out of the cell. But the same thing can happen otherwise. Something outside of the cell can attach to the plasma membrane. We can wrap the plasma membrane around it to make a vesicle and actually bring it into the cell. And what would we call that process? There you go. Devlin beat me to the punch. Endocytosis. Excellent. So endocytosis to bring something in, exocytosis to bring something out. So we can use those vesicles to bind and release or to bind and bring in. And for the record, those are active. They do require ATP. But they allow us to get a lot of stuff or really big things into or out of the cell. All right. Questions on that? All righty, excellent. So we did that, All right, so let's go back to our beaker with my saran wrap down the center. And again, when I do something really sophisticated, like taking a screwdriver and poke a hole in that saran wrap, right? Does that hole determine what direction my molecule is going to go?
No.
No, if I have 45% Kool-Aid on this side of the beaker, do I know which way it's going to go?
Depends on what's on the other side.
Exactly. It depends on what's on the other side. If on the other side it is 75%, oops, 75%, which way is it going then?
Both.
Well, it will go in both directions, but what is the net movement going to be?
Left.
Net movement is going to be left. Excellent. And it will continue going left until what happens?
Equilibrium.
Equilibrium. Well, what would equilibrium be at this point?
60%. Both sides?
Exactly. 60% on this side, 60% on this side. At that point, does the movement stop?
There's not going to be any net change after that point.
Exactly. Movement still continues, but remember, at equilibrium, there is no net change. Right? So notice my holes don't determine which way the molecule moves. The concentration gradient does. And it's the same thing that's true for these passive channels, right? And again, just to be clear, when we talk about a channel and a pore, essentially those terms are interchangeable. Let's cheat and do it here. Here, get rid of this just so it's out of my way. Channels are formed by proteins that are embedded within the plasma membrane. So these proteins form the channel. Of course, there is a hole in the channel that stuff passes through, and that hole in the channel is what we call the pore. So when we talk about a channel or we talk about a pore, we're basically talking about the same thing. The channel is the proteins that make the hole. The hole itself is the pore. And the point of this is that they are like holes that is punched with a screwdriver, like the door frame of your door to the room that you're in. That door frame doesn't determine which way you go into or out of the room, right? Your motivation determines which way you move into or out of that door, right? It's passive. But notice, it can also be closed and locked. It can be gated. It also can be restrictive. You can get in and out of that door, but could an elephant get in and out of that door? Could you park your car in your bedroom if you wanted to? No. So again, these channels, while they're passive, can still be selective, and they can also be gated. They can actually be opened and closed as well. So they're a little more sophisticated than just a hole punched in the plasma membrane, but they're passive and they don't determine which way things move. As we've talked about, molecules are going to move from a high concentration to a low concentration. They do not require any energy from you. And the classic example of this is if I'm standing at the top of a hill with a ball, that ball has a lot of built up potential energy in it because it's at the top of the hill. So when I let it go, I do not have to put any energy into that ball. That ball is going to roll down the hill on its own. And just like we talked about, the steeper the concentration gradient, the faster that ball is going to roll. So things move from a high concentration to a low concentration. However, if I want to get that ball back to the top of the hill, do I have to use energy then?
Yes.
Yeah, absolutely. And so if I want to get a molecule to move from a low concentration to a high concentration, I am going to need to use energy. And I'm going to need to use a special protein. And that special protein is either called a transporter, or we also sometimes refer to them as pumps. So it's going to require a special protein. It's going to require the use of energy. And again, remember, when we use and talk about energy in the cell, we are talking about ATP. We're going to need to use a special protein, a special transporter or pump that will take that energy and push that ball back up the hill, move that molecule against its concentration gradient. All right. Questions on that? All right, excellent. All right, so as we talked about before, one of the main ways we get cells to do work is by having an unequal distribution of ions inside and outside of the cell. And we can give a fancy name to this. We can say it is a chemical gradient. And it allows us to do, or let's say it this way, it produces a chemical force. So there were four main ions we talked about. Remind me what those were again. Give me one of them.
Potassium
Potassium, excellent. Where was there more potassium?
inside the cell.
Excellent. So we know that there was more potassium inside the cell. Give me another one.
Sodium.
Sodium.
Where was there more sodium?
Outside.
Outside.
Outside the cell. Give me another one.
Chlorine.
Yep, where was there more of that?
Outside.
Excellent, more chloride ions outside the cell. Oops. And what was the fourth one?
Calcium.
Calcium. Where was there more calcium?
Outside.
Outside.
Excellent. Excellent. So we did that. Less potassium. Oops. Outside. Sodium inside. Less chloride inside. Less calcium inside. Excellent. So based on our chemical force, based on our concentration gradient, which way does sodium want to go? Into the cell or out of the cell?
Into the cell.
Into the cell, absolutely. So what we can say is that there is a chemical force or concentration gradient that is driving sodium into the cell. All right. What's the chemical force? What's the concentration gradient for potassium?
Out.
Out. What is it for calcium?
In.
What is it for chloride? In. See how easy that is? So just that quickly, we have identified the chemical forces for all of our four major ions. But there's one other important thing to remember about these ions, and that is that they're ions, right? And what does that mean to be an ion?
They have a charge.
They have a charge, absolutely. So while it is 100% accurate and true that there is an unequal distribution of ions inside and outside of the cell, it also turns out that there is an unequal, let's make this bigger. And let's make it black. Distribution of charge inside and outside of the cell. Right, because these ions have a charge to them, there's an unequal distribution of charge inside and outside of the cell. We actually call this the membrane potential. And essentially, this membrane potential produces an electrical force. Now, it turns out there's an unequal distribution of charge in the ion. And as it turns out, the inside of the cell is more negative than the outside. And we can actually put a number on it, right? The resting membrane potential, right? Membrane potential when the cell is not doing any work. So the resting membrane potential of the cell is negative 70 millivolts. Meaning that if you leave the cell alone, it will happily stay at negative 70 millivolts forever. Now, this is a negative charge. And as we know, anybody who's played with magnets before, do positive things like negative things, or do negative things like other negative things?
Positive. Like negative.
Positive like negative. Excellent. So if we were to talk about sodium, for instance, based on sodium's electrical force, which way do you think sodium would want to go? Just based on its charge. Does it want to go inside the cell or does it want to go outside of the cell?
Inside.
Inside, absolutely. It wants to go in. In fact, sodium really, really, really wants to get into the cell. Sodium's huge divide, notice its chemical force, its electrical force, it really, 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 really wants it to get to the cell. So a lot of times when we need to do work, we open a channel and let sodium come in and do that work for us because it really, really, really wants to go inside. Now notice calcium also really, 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 really wants to go inside. Its chemical force and electrical force are both inside. But why don't we necessarily want calcium coming inside the cell all the time?
Really positive.
True, it is really positive, but if you also remember, I told you that calcium makes cells do wonky things. All right. If you want to produce an action potential, if you want to contract a muscle, if you want to release a secretion, right? And do you necessarily want to be doing wonky things all the time? No. So we're going to be a lot more selective, even though calcium really wants to go in, because calcium makes cells do wonky things. And yes, that is the technical term for it. We are going to be a little more selective about how we let it in. So most of the work we're going to do with most cells is going to start with sodium. Notice for chloride and potassium, it's a little bit more complicated, right? Potassium is a positive ion. Its concentration gradient wants it to move out. But what about the electrical force of potassium? Which way does the electrical force of potassium want to go?
In.
It wants to go in. Now, what do you think? Are these two forces equal?
No.
No. You know the answer to this. When you open a potassium channel in the cell, which way does potassium go? Does it go out of the cell or does it go into the cell?
In.
Out of the cell.
Out of the cell. So the chemical force is bigger. So there is indeed an electrical force on it, but the chemical force is bigger. So, whoops. So when we open a potassium channel, potassium does go out, but there is an electrical force driving it in. Chloride also has an electrical force. And being a negative ion, which way does its electrical force want to drive it?
Out.
Yeah, it wants to drive it out. So again, two competing forces. And not surprisingly, which one do you think is bigger?
Chemical.
Chemical, yep. So when you open a chloride channel, basically what happens is chloride comes in. Okay. The take-home message of all of this is just gradients plural. We can't forget about that electrical force when we're talking about this. But in a resting cell at its normal starting point of negative 70 millivolts, basically this is how all of our ions, these two forces determine how all of our ions move. If you open a sodium channel, sodium's going to come into the cell. If you open a calcium channel, calcium's going to come into the cell. If you open a chloride channel, chloride's going to come into the cell. And if you open a potassium channel, potassium's going to leave. And just to sum this all up, let's think in terms of the effect. If sodium enters the cell, what kind of an effect does that have on the cell?
Makes it more positive.
Yeah. If chloride enters the cell, what is its effect going to be?
More negative.
If calcium enters the cell, what's the effect going to be?
More positive.
Excellent. And it's going to make the cell do wonky things. And last but definitely not least, What is the effect of potassium leaving the cell?
More negative.
There you go. When a positive ion leaves the cell, it makes the cell more negative. Excellent. Questions on that? All right, excellent. Stunned silences mean, of course, that you understand everything. So let's add one more piece to this puzzle. These ions are constantly moving. Sodium is constantly coming in, potassium is constantly leaving. And let's think about that. This cell has a resting membrane potential of -70. If one positive thing comes in and one positive thing leaves, is that going to affect the resting membrane potential? What do you think? If I give you $1 and you give $1 to your kid, are you more rich or less rich than you were before?
The
No.
same.
You're the
The
same.
same.
There's not going to be a change. However, remember, we need these ions to move to continue to do work. So we have to have a way to maintain this. If sodium is going to constantly sneak in and potassium is going to constantly sneak out, the membrane potential of our cell isn't going to change, but our concentrations of our ions will. And if the concentrations of our ions change, they may not want to do work for us anymore. And that would be a bad thing. So luckily, we have a very special pump. And this very special pump's job is to use ATP. And it uses that ATP to do some work for us. It is going to move sodium. Now, if we're going to use ATP, which way do you think we're going to move the sodium? Into the cell or out of the cell?
Out.
Out, right? Because it wants to come in. So, oops, that's way too big. It wants to come in. So if I'm going to use ATP, it's going to be to kick sodium out. And not just any old sodium, but I'm going to be able to use it to kick 3 sodium out of the cell. However, at the same time I'm using this ATP to kick 3 sodium out, I'm also going to be moving some potassium. And which way do you think we'll move the potassium?
In.
In. Exactly. And in this case, it actually only brings 2 potassium in. Oops. Out. So 1 ATP brings 2 sodium in and kicks 3 potassium out. This very special transporter we call the sodium potassium ATPase, because it's an enzyme that breaks down ATP to do work, or it's also sometimes just simply called the sodium potassium pump. When a cell is at rest, about 25% of its ATP is used just by this sodium potassium pump. Because what's happening is sodium is constantly sneaking in, and when it sneaks in, we have to kick it out. And potassium is constantly leaving, and so we have to bring it back in. So when a cell is at rest, not doing any other work, it's still very active, and about 25% of its ATP is just used to maintain this sodium potassium concentration. And notice too, when this pump is working, it kicks 3 positive things out. How many positive things does it bring back in?
2.
Only 2. Notice, so again, remember its resting membrane potential is negative, and this pump plays an important role in maintaining this negative. No, calcium has 2 charge. What I'm saying is there are actually 2 potassium that are being kicked out, the same way that there are 3 sodium being brought in. So yes, so 3 positive things are kicked out, 2 positive things are brought in. Helps to keep the cell negative. Calcium, again, because it can basically donate 2 electrons, has a positive 2 charge. Sodium and potassium only have a positive 1 charge. So great question. Yep. All right, excellent. So I've done a good job of describing this. Let's go ahead and clear it. And again, do the same thing. Here we see that chemical gradient, that unequal distribution of ions. Here we see that electrical gradient, that change in the membrane potential where it's negative inside and positive outside. And again, collectively, they form that electrochemical gradient that affects which way ions move. All right, questions on that? All right, we've talked about the movement of ions. We got to talk about the movement of water next, but let's go ahead and take our first break. We're doing really good on time. Looks like it is 9:08. So any questions on this before we take our first break?
I just have a question of when the videos of the lectures will be uploaded. Will they be uploaded by this weekend by chance?
It should be. I will warn you, I am going away this weekend, so I will be out of town this weekend. I do know, and I appreciate you asking this question. Usually it's within the next 24 hours. I saw this morning that that yesterday's lecture hadn't uploaded properly to YouTube, so I submitted it again this morning before class. So my guess is by the end of class today, yesterday's lecture should be up.
Okay, and then, oh, sorry, go ahead.
Well, I was gonna say, I will attempt to upload the video today before I leave for the weekend, but there is a slim chance that if there's another problem, it won't be up till Sunday when I get back. But I'm hopeful. My goal will be to try to do it beforehand, but if there's any problems, I won't be around to resolve them.
That's fine. And then I was curious, is there a link in Canvas that we can click to be able to watch the lecture videos, the past ones? Or
Yeah, I believe it's under the study tools. Under the study tools,
okay,
it's linked to my YouTube channel.
Okay,
yeah,
thank you.
You probably don't want to go to YouTube and type in Slutsky because who knows what will come up if you do that. But there is a link directly to my page from the Canvas site.
Awesome,
right.
Thank you.
Yep, excellent. All right, any other questions? Oh, and of course, remember to smash that like and subscribe. All right. All right. I don't care. Any other questions before we take our first break? All right, excellent. It is 9:10 now, so let's take a 15-minute break, come back at 9:25, and at 9:25, we will pick up from there. All right, see you guys in 15 minutes. Let's go ahead and get started. So we have just finished talking about the driving forces on solutes. But it is important to remember that there are also driving movements on the solvents as well. And of course, the primary solvent that we're talking about when we're talking about biology, when we're talking about anatomy and physiology, is water. All right. Now, as I mentioned, osmosis is a big fancy term that many of you have heard of before, but it really is just the simple diffusion of water. And the good news about that is it follows the same rules, right? If, again, you've ever taken like a biology 300 class or something along those lines, and we have our beaker. And as we talked about, we'll use the example we had before, 75% glucose on this side. Make that a little smaller. And 45% glucose on this side. On this side, as we've just finished talking about, when I poke the holes in the bladder, which way is glucose going to move?
The right.
Yeah, it will move to the right, and it will continue to move to the right till it reaches equilibrium. And what did we say equilibrium would be in this case?
60%.
Yeah, it's going to be 60%. So it'd be 60% on this side. Oops, that's not a percentage side. 60% on this side, and 60% on this side, at which point it would be at equilibrium, right? But one of the important things to remember is when I say that this is 75% glucose, what's the other 25%?
Water.
Yeah, this is 25% water. Right? If the right side is 45% glucose, then what's the other 55%?
Water.
Water. Excellent. Oops. There we go. It's going to be water. And water moves just like solutes do. Right? If there was some type of special receptor within the walls of this saran wrap that didn't allow that big molecule that was a glucose to move, but did allow water to move, which way would water move?
Left.
Yeah, water would want to move to the left. Water would also move down its concentration gradient. Right, like I said, if you took a biology 300 or 400 class, one of the things that you hear is water follows salt. Water goes to where there's more stuff. So water follows salt, or water follows stuff. And if those are the rules you've learned and that's the way you want to think of it, I'm not going to change your mind, and that's great. We know it's going to move from the right to the left to go to where there's more stuff. However, I don't like new rules. And it's just easier for me to remember that water also moves down its concentration gradient. And it will move from a high to low. And it'll continue to move high to low till it reaches equilibrium. And what would the equilibrium point be in this particular beaker?
40-40.
Yeah, it's going to be 40-40, exactly. So it would be at this point 40% water on this side and 40% on this side. Now, obviously, it takes a special beaker, and you just can't put saran wrap through it to get this to work. But this is exactly how water moves. This is exactly the driving forces on water. And in fact, if we go ahead and erase my simple drawing here, we can take a look at the type of mechanism that is used for this. Back in ancient times, and by ancient times, I mean when I was in grad school, this is what we meant by U-tube. Right? It was a U-shaped tube that had a special diaphragm in the center of it. Notice they have the same amount of water on both sides, but on one of these, they've put a lot more glucose than the other. The diaphragm on this is specialized with a small pore so that glucose cannot move through. So yes, there is a driving force on glucose. Glucose wants to move to the left, but it cannot. But what can move is water. And so water will move from the left to the right, not till the volume of water is the same, but until the relative concentration is the same, just like we saw. So 75 on one side, 45 on the other of our glucose, right? And water moves, right, so as we saw, or in this case, it would be 55% water on one side, 25% water on the other side. It moves till it's equal, 40-40 on both sides. Notice the volume's not the same, it is the concentration that is the same. I like this one because it's a simple illustration, just shows the glucose molecules, but you'll notice, and I think this is the one from your textbook. I also like this one. This one isn't from your textbook. What they do a nice job showing is that the water molecules are smaller than the glucose molecules. So again, water goes down its concentration gradient from a high concentration of water to a low concentration of water until the relative concentrations are the same. This is osmosis, and that driving force of water from the left to the right is what we call the osmotic pressure. Now, once my U-tube looks like this, is there any way to get that water to go back to the other side? Is there an opposite driving force of osmotic pressure?
Yeah.
Yeah, I'm asking the question, so the obvious answer is yes. So what should it be? What is the, how would I get water to go back to the other side?
Add salt.
Say again?
Add salt.
Okay, true, you're right. I could change the osmolarity by adding more solutes, but is there a way that without changing the solutes, I could move the water back to the other side?
Force it down.
Force it down, absolutely, right? I could put a force on the water, like for instance, putting a weight on this side. And by putting a weight on that side, I can put a force on the water. And of course, we have a fancy term for that force on the water. We call it a hydrostatic pressure. So the opposite of osmotic pressure is hydrostatic pressure, the force on the water, the force we're putting on the water, or the force of the water on its boundaries. Right, and that's how we define that hydrostatic pressure. It is the force on the water, or we could think of it as the force of the water on its boundaries. Of course, the question becomes, why do we care? Right? Why are we bothering to learn these things? Why is it important? Right? Well, back in ancient times, and by ancient times, I mean last year, when it was about this time of year, it was about 110 degrees outside. Right? I know when I went for my bike ride this morning, I needed both a long shirt and a jacket because it was so dang cold. But back in ancient times, the temperature used to get above 100 here in Sacramento. Who knows, maybe it will again someday. I can't tell anymore. But if it is 100 degrees outside, is that necessarily the best time to go outside and tar your roof? No, probably not. And if you did do that and spend about 6 hours tarring your roof on a 100-degree day, you would probably dehydrate and be rushed to the hospital. And when you got rushed to the hospital because you were dehydrated, do they take a just big, huge bottle of deionized water and slap it right into your arm?
Yeah.
Really? Deionized water?
No.
No, absolutely not, right? That'd be a bad thing, right? The reason for that is what we call tonicity. We know that the cells inside of our body have water inside of them and have stuff. So we have water and stuff inside of our cells. And they need to be surrounded by water and stuff. Right? And the water and stuff that surrounds a cell, we want to be equal to the water and stuff inside of the cell. If, like I said, we just put deionized water, into here, what percent water is deionized water? What? Okay, let me ask the question this way. What else is in deionized water besides water? None of you heard of deionized water before?
Water.
Well, again, it warms the cockles of my heart when you guys don't know chemistry stuff, because like I said, chemistry is stupid. But the simple answer to that is it's 100% water. So if it's 100% water, how much stuff does it have in it?
0.
0% stuff. All right, is that more stuff or less stuff than's in the cell?
Less stuff.
Excellent. So now we have an unequalness between what's inside the cell and what's outside the cell. And as we know, water goes to where there's more stuff, or water moves down its concentration gradient. So if I were to pump a liter of deionized water into your blood, or if you were to drink a massive amount of water in a very short period of time to win an antiquated video gaming system, you set up an environment where there's not enough stuff around your cells, water rushes into your cells, and your cells swell. And like a water balloon, if you put too much water into that water balloon, what happens?
It bursts.
It can burst. So not only can it cause the cell to swell and stop functioning normally, but it also can cause that cell to burst, causing that cell to die. leading to cell death, leading to organ failure, leading ultimately to death. Right? We get the cell to swell, and that cell, the term we use for it is it lyses as a result of that. And this type of an environment is what we would call a hypotonic solution. All right? Instead, if we wanted to, if you rushed you into the emergency room because you were dehydrated, we wouldn't just put deionized water in there. What would we give you?
Saline.
Yeah, some kind of saline or some kind of lactated ringer that has the same amount of water and stuff inside of it as in the cell, at which point, right, there is no net movement, there is no net change, and this would be what we called an isotonic solution. But let's think of a third example. In this third example, you are out fishing this weekend because you got the nice big weekend, and you're out to sea looking for those marlins, and your boat capsizes. When that boat capsizes, obviously your number one concern is going to be sharks. But as you start reaching day 2, day 3, day 4, just hanging on to the edge of that boat, completely submerged in salt water, is that necessarily a good position to be in? No, you might get thirsty. You could always drink the water around you, right?
No.
You wouldn't want to do that. And just being exposed to that salt water is a bad thing, because that salt water of the ocean has more stuff in it than our cells do. We would call this a hypertonic solution. And what happens if the salt water has more stuff into it? What actually happens to our cells?
They shrink.
Shrivel up.
Yeah, water is pulled out of those cells, and those cells shrivel up. Right? They actually do this with red blood cells. In fact, the example that I'll show you, they do this kind of experiments with red blood cells all the time. And that crumpling up of the red blood cells is a process they call crenation. So the environment that we put our cells in is vitally, vitally important, because this movement of water affects the health and the function of these cells, affects their tonicity. Again, I've done an amazing job of drawing this, but let's look at the pretty pictures from your textbook. Here we see an example of that happy red blood cell in its isotonic solution. Hypertonic solution, which way does water move in a hypertonic solution again?
Out.
Out of the cell, causing the cell to crumple up in that crenation. And that means in a hypotonic solution, water goes in, causing that red blood cell to swell up like a big balloon. And if it swells up too much, it can actually rupture and burst. That cell can lyse. All right. Questions on that? All right, excellent. So notice what we've been talking about are these concepts of membrane transport. Now that we have an understanding of the plasma membrane and how the plasma membrane works and how things move, we need to talk about this concept of membrane transport. Now, remember, we also already talked about this concept of passive versus active. What does it mean to be passive again?
No
Does not
energy.
require energy.
And again, let's be a little bit more specific. It does not require ATP, right? Technically, it is using the kinetic energy of the molecule. So it isn't so much that it doesn't need energy, but what it doesn't need is ATP. There's no ATP used for it. And because we're not using ATP, what is the primary rule of how things move?
Diffusion.
Right, diffusion, true, diffusion, but directionally.
Higher concentration to lower concentration.
From high concentration to low. Excellent. How does that compare to active transport?
The opposite.
Yeah, absolutely. Right. With active transport, either directly or indirectly. Oops. Uses ATP. And if we're going to use that ATP, typically what direction are we trying to get something to move?
Low to high.
Yeah. Excellent. So when we're talking about this, we're talking about the use of ATP. We are talking about these processes. Now let's actually do this on the whiteboard. I like this on the whiteboard. All right. Get there. There's my plasma membrane. And for starters, let's go here. I think that should be enough space. And as we talked about, we'll do over here, passive membrane transport. I'll make that a little bit bigger. And as we know, with passive membrane transport, does not use ATP, and it goes from high to low concentration. All right. No new information there. I've just moved what we talked about before. So hopefully we're comfortable with that. Yes? Excellent. My stunned silences, of course, mean yes. So perfect. All right. Now, as we've also talked about, there are different types of passive membrane transport. The most basic of this passive membrane transport, as we've talked about, is just simple diffusion. With simple diffusion, someone remind me again, what type of things typically move through the plasma membrane via simple diffusion?
Small, nonpolar, lipid base.
Excellent. Things that are small, things that are nonpolar, things that are lipid based, they can merely just pass through the plasma membrane without any help at all. Is there anything else that's able to move via simple diffusion?
Water.
Yeah, remember also we talked about water. Oops, what just happened? Water also uses simple diffusion, but we do give a fancy name for the simple diffusion of water. And what was that fancy name for the simple diffusion of water?
Osmosis.
Osmosis. Excellent. So those are the things that can pass through the plasma membrane without any help at all. Other things then require the use of a protein. And if it requires the use of the protein, it is what we call facilitated diffusion. So facilitated diffusion is where we use a protein but no ATP to move things across the plasma membrane. And there are basically two different types of facilitated diffusion. The first are what we've already talked about, are channels. And remember, when we talk about channels, basically we're also talking about pores. And those channels, those pores that we've talked about, are, like I said, like your door frame. Basically what we have are, well, here, let's cheat and do this first. A bunch of proteins that form an opening, and that opening is the pore that allows substances to get through, right? Your door frame allows you to go through whichever direction you want. right? But as I mentioned, it has a lock, it has a door on it that has a lock. So it can be open sometimes and it can be closed sometimes. It can be selective, where it only allows sodium through, but not potassium, right? You can get through your door, but an elephant couldn't. So it still can be selective, it still can be opened or closed, but it does not change shape. The frame of your door doesn't change shape when you pass through it. There are some things that can change shape when you go through them, and those things are called carriers. Now, I know these can sometimes be hard to imagine, but they change shape. But let's see this way. How many people here have ever gone to the Sacramento Zoo before? Anybody go to the zoo before?
Yep.
Excellent. How do you get out of the zoo?
Oh gosh,
Say again?
it's been a long time, but the exit.
Okay, anybody remember what the exit's like at the Sacramento Zoo? I know with COVID, nobody's been outside of the house for a while. It's more than just a gate. No one remembers? No one's got kids?
Like
No
a,
one's...
it's like
Say
a
again?
turnstile. It's like a turnstile
It's a
gate
big,
thing.
huge, massive turnstile. Absolutely. Now, as you walk up to that turnstile, does the Sacramento Zoo have that plugged into an electrical outlet where they're producing the juice, they're producing the energy to keep that door revolving, and you have to just walk through it?
No.
No,
No. If you want to get out of the zoo, what do you have to do?
manually push it.
Yeah, you have to push on that revolving door so that it changes shape so that you can get out. And that is exactly what a carrier is. Basically, a carrier changes shape, but it uses the energy of the molecule. It is not using ATP. This is still a passive process. Now, this is typically used for larger molecules like glucose. Glucose is a great example of this. Glucose is a really big molecule. So if you just had a channel in your cell that would allow glucose to pass through it, all sorts of other stuff could get in and out as well. And that would be a bad thing. We don't want that. So instead, what happens is we have this special carrier. And this special carrier basically is open on one side. So here's one of the proteins. Here's the other protein. And it's open on one side. And what happens is our glucose comes in and binds to it. And when our glucose comes in and binds to it, I think I'm going to cheat and have to give myself a teeny bit more room. When the molecule binds, it, there's that fancy term again, undergoes a conformational change. And someone remind me again what that means? Come on, what does it mean to undergo a conformational change? It's a fancy way of saying what? All right, did my microphone just suddenly go silent?
Did it just change according to its environment?
Well, it just means it changes shape.
Okay.
It's just a fancy way of saying that it changes shape. And when it undergoes a conformational change, it changes shape. And so notice when it changes shape, what ends up happening is the top closes and the bottom opens. And that allows the glucose to enter into the cell. And once the glucose lets go, guess what happens?
It goes back to its original.
It goes back to its original shape. And then a new glucose can bind to it, it changes its shape again, and it brings in that glucose. And then it goes back to its original shape. Again, notice the cell is not using any ATP to do this. It is just using the energy of the molecule. That is why this is still a passive diffusion, because no ATP is being used. All right, and so those are the two types of facilitated diffusion, our channels and our carriers. All right. Questions on passive membrane transport? Again, if you don't like my drawings here, we've got the pretty pictures from your textbook. Again, with simple diffusion and facilitated diffusion, channels and carriers. Here is the pretty picture from your textbook showing a channel. Notice theirs is a little nicer than mine. Again, here we see our phospholipid bilayer. Here we see the proteins making the channel. And again, the hole in it is the pore. Notice it can be selective. This one just lets potassium leave the cell. Notice this one also can be gated. It can be open and closed. These are two special proteins. Notice one is a big globular protein and one is a long linear protein. They actually have a very technical term for them. These are referred to as the ball and chain. And notice this ball and chain can wedge itself into the channel. And when it does, the channel's closed and potassium can't move through it. So again, these can be selective, they can be specific, they can be gated, but they're not using any ATP and they don't change shape. This is different from our carrier. Notice again, a little bit better drawing, our phospholipid bilayer. Here are our proteins. Notice they are open at one end. Do not use ATP, they're passive. And this allows larger things like glucose to get in. When that glucose binds, our protein undergoes a conformational change, which is a fancy way of saying what?
Changes shape.
Changes shape, absolutely. Right? And when it changes shape, it opens at the bottom and closes at the top, allowing the glucose to come in. And then as we mentioned, once the glucose lets go, it goes back to its original shape and a new one binds and it brings it in. And then a new one binds and it brings it in. And a new one and back and forth, letting the glucose into the cell, but just using the energy of the glucose, not using any ATP. All right. So questions on our passive membrane transport mechanisms.
Is it just like going like off the weight of whatever it is, or is it actually like drawing something out of it? Or,
Great question. There's typically two things that causes those change. The first of them is the kinetic energy of the molecule. So sometimes they will use the kinetic energy of the molecule to change their shape. But the other thing about it is hydrogen bonds. Remember, many of these molecules have functional groups that can be positive or can be a negative. And so the attraction and the repulsive, those hydrogen bonds with those functional groups can also be what causes it to change shape as well. So the same way that, a magnet pushes, a positive magnet pushes another positive magnet away, those kind of electrical forces can be what's being used. So it's not using ATP, it's just using the kinetic energy of the molecule and the attractive and repulsive forces of the functional groups to change its shape. Yep, great question. Any others? All right, excellent. That means we understand. How are we on time? We're doing good. Perfect. Understand means we understand our passive membrane transport. So then let's talk about our active membrane transport. Excellent. Remember, with our active membrane transport, we are either going to directly or indirectly use ATP. We are always using a protein. And since these always use proteins, these are all facilitated. Right? Because again, facilitated just means that it is using a protein to get some help. And remember, with active transport, we are typically moving this against the concentration gradient. So from low to high. But notice twice now I have said either directly or indirectly uses ATP. because as it turns out, there are two types of active membrane transport. There is primary active transport, and there is secondary active transport. With primary active transport, this one directly uses ATP, and secondary active transport indirectly uses in ATP. And we'll explain what that means in just a second. Our primary active transporters, these are things that we typically think of as our pumps. They use ATP directly to move a molecule. We've already talked about one of these, right? The one we talked about already was that sodium-potassium ATPase, or again, what we call the sodium-potassium pump. And again, it is directly using ATP. So an ATP molecule binds to it. And someone remind me, when it phosphorylates that ATP, uses the energy of the ATP, someone remind me again what it moves?
2 sodium out, 2 potassium in.
All right, so 2 potassium come in. How many sodium leave?
3.
3, excellent. So it kicks 3 sodium out and brings 2 potassium in, right? So that was an example. And there are even more basic examples, right? Well, as I mentioned before, calcium makes cells do wonky things. So not surprisingly, massive numbers of our cells have calcium pumps. And calcium pumps are, of course, going to use that ATP to move calcium. And guess which way we want to move the calcium?
Out.
Yeah, we want to kick that calcium out. So we're going to use ATP to kick calcium out of the cell. So these are our examples, and let's cheat a little bit. Oops, didn't want that. Use a little bit of our gray highlighter here to separate that. Wow, that's way too big. I don't need that to be that big. Perfect. So 2 examples of our primary active transporters are pumps, where we are directly using ATP to move materials into or out of the cell. All right. Questions on that?
Nope.
Excellent. So let's talk about a secondary active transporter. As I mentioned, it indirectly uses ATP. It doesn't use ATP directly. So let's see an example of this. Now, remember, one of the things that we said is sodium really, really, really wants to get into the cell. So what I do is I say, okay, sodium, you can come in. Go ahead and come in. Come on in. That makes you happy. But when you come in, I'm going to use you to do some work. Think of it this way. We use the example of me standing at the top of the hill with a ball. If you were at the bottom of the hill and I had just finished your grading your exam, I could walk that exam all the way down the hill to give it to you, but that would require a lot of energy on my part. Instead, I could tape it to that ball and let the ball go and let the ball roll down the hill. And when it reached the bottom of the hill, you would get your exam. And did I have to do any work? No, I let the ball do it for me, right? Or again, this is the exact same concept with water wheels, right? Back in ancient times, if you had a mill, or if you wanted to, mill flour, if you wanted to, chop down the trees and have a lumber yard or something like that, you built that right on the edge of a river. And you put a big paddle wheel into the water. And as the water went down, it spun your wheel, and you could use that energy to do work, to grind your flour, to saw your lumber, to do whatever you wanted, right? We're letting sodium move, and we're getting it to do some work for us. All right, sodium, come on in. But when you come in, you know what? You need to bring an amino acid in with you. Or you need to bring a glucose in with you. Something like that. Notice these secondary active transporters are also known as co-transporters, because they're moving multiple things. Now, notice nowhere in here did I put ATP. So how is this an active process?
Is the co-transporter using ATP?
But I didn't draw an ATP anywhere on here. That would be a good guess, but it's not. But think of it this way. We are able to do work because sodium wants to come in the cell. Let's say, for instance, and again, these numbers obviously aren't accurate, but let's say, for instance, there were 10 sodium outside the cell, and there were 6 sodium inside the cell, right? Sodium would want to come in, and so I could get a sodium to come in, and it would bring a glucose in with it. But then how many sodiums am I going to have outside after that?
9,
9. And how many sodiums am I going to have inside after that?
7,
And so a sodium comes in, and it brings in an amino acid with it. And how many sodiums do I have outside now?
8,
And how many sodiums do I have inside now?
8.
Does sodium want to come inside anymore?
No.
No. And if sodium doesn't want to come inside anymore, can I get it to do work for me anymore?
Nope.
No. So notice in this case, again, the key is I'm indirectly using ATP. This molecule itself doesn't use any ATP. But as soon as that sodium comes in, what do I have to do?
Kick us 3 out.
I have to kick it out. Exactly. So in this key, I allow an ion in. And again, that ion is usually sodium, because as we said, sodium has a huge driving force wanting to come in. There's sometimes we use other ones, but most of our co-transporters use sodium. We allow sodium to come in. It brings something else in too. It does work. But then we have to kick that ion out. That sodium back out. And what do we need to use to kick that sodium back out?
ATP.
Yeah, we have to use ATP for this. So this is how it is indirectly using ATP. The molecule itself doesn't use ATP, but if I don't use ATP to kick that sodium out, sodium's not going to want to come in anymore. Now, notice this one here, the sodium and the amino acid and the glucose are moving in the same direction. And since they're moving in the same direction, we call this a symporter. But it works the other way as well. I could have a molecule that, again, lets sodium in, because sodium really, really, really wants to come in. And I say, okay, sodium, you can come in, but when you come in, I need you to kick out a calcium, because as we said, we don't want calciums in the cell. or I need you to kick out a hydrogen ion so that my cell doesn't get too acidic or something like that. In this case, my substance that is being moved moves the opposite direction of our sodium. And so this is an example of what we call an antiporter. So our secondary active transporters are co-transporters, either symporters or antiporters, moving things either in the same direction of sodium or the opposite direction of sodium. And those are our different types of active membrane transports. With one more exemption. Don't forget about vesicles, right? We can have that vesicle that binds and releases that substance, right? Releasing things this way or bringing things in. So again, remember the last type of active transport is our vesicle transport. Right? Endocytosis or exocytosis. So those are our final type of, again, that does require ATP as well. We don't need to know the mechanism, but using vesicles to get things in and out is our last form of active transport. So I guess we need to draw a third line here. So, way too big. Sneak it in that way. There we go. So primary active, secondary active, and then vesicle transport are three types of active membrane transport. And again, I've done an amazing job drawing this, so I'll actually post this on our website. But let's take a look at the pretty pictures from your textbook. Again, with our active membrane transport, are we on time? Good. Again, it's mediated or facilitated. Either of those are the terms we can use. But remember, it all means the same thing. Basically, this just means uses proteins, right? There is no simple active transport. If you're using ATP, you have to use some type of protein to get things through. Right. Again, with active transport, typically we're moving things where they don't want to go. So that's usually against their concentration gradient. There will be some places, like for instance, when we talk about the urinary system or digestive system, where we're going to use active transport because we really want to get the things where we want them to go. But usually it's against the concentration gradient. And again, it comes in two flavors, primary and secondary. Secondary active transport are our co-transporters. They indirectly use AT. They do not use ATP directly, but we need them to pump them out. And remember, there are two types of secondary active transporters. Symporters, where both ions move in the same direction. And antiporters, where both ions move in opposite directions. Excellent. Again, no new information there, just stuff we've already seen. And again, the whole point of this was to show you the pretty pictures. Here is actually your friend and mine, the sodium potassium ATPase. And again, if you haven't figured it out yet, you are going to know it eventually because we're going to talk about it a ton. Notice here it is using the energy, phosphorylating the ATP, breaking off that phosphate and using the energy to change its shape. Notice first it's open to the bottom, to the inside of the cell, which allows 3 sodiums to bind to it. Then it uses the energy of the ATP to change its shape, expelling the 3 sodium and allowing 2 potassium to bind. When those 2 potassium binds, it kicks off the phosphate, goes back to its original shape, and our potassium comes in. Then 3 sodiums combined, use the ATP to change its shape, and this process continues. So 1 ATP kicks 3 sodium out, and brings 2 potassium in. Here are some simple illustrations of our secondary active transporters. Notice here, sodium comes in and kicks out of calcium, or sodium comes in and kicks out of hydrogen. These are our antiporters because they're moving in opposite directions. Here, sodium comes in and brings a glucose with it. Sodium comes in, brings an amino acid with it. These are moving in the same direction, and these are our symporters. And again, we're not using ATP directly. The whole reason this works is because sodium really, really, really wants to come into the cell. So as soon as that sodium comes into the cell, we then have to use ATP to kick it back out so that these co-transporters will keep doing work. All right. Questions on that?
And these are all the integral proteins.
Yes, and more specifically, notice transmembrane integral proteins, because notice they have to go through both layers of the plasma membrane if they're going to bring something in or kick something out. So yes, all of these are integral. All of these are transmembrane integral proteins because they have to go through both layers or they won't be. And the same thing's true for your carriers and for your channels. If they don't go through both layers of the phospholipid bilayer, they won't be able to let things in or out. So notice all of these things, yes, absolutely. Not just integral, but transmembrane proteins. Got to go through both layers. Excellent, great question. Any others? All right, excellent. Then that is all I have from a lecture standpoint for you for today. All right, so let's talk about the lab exam. Let me get this set up. Oops, didn't mean to do that. All righty, so let's actually go here. I think I saved this, but let's save it again just to make sure. And let's go ahead and clear this and talk about what we're going to do. All right, so you are going to have 30 minutes to take the lab exam. All right, there are 20 questions on it. Usually you get about a minute a question for the lab exams and about 5 to 10 minutes. So 20 questions, 5 to 10 minutes for all the procedural stuff, for the proctorio stuff. The showing of the ID, the scanning of your room, and everything else that goes along with that. All right. You also, I encourage you to take a 15-minute break. You can either take that right now and then take the quiz, or you can take the quiz first and then use the rest of the time. But either way, you must be done by 11:30, and we will re-meet to discuss the exam at 11:30. So at 11:30, make sure you are done and be back here at 11:30. So again, that gives you, well, now about 10 minutes of a break. Sorry, hold on. No, it's 10:20, so half an hour plus 15 minutes. So let's make it 11:15. That's more than enough time to take the exam, more than enough time. So let's fix that. So we'll do that 11:15. So I gotta go back and change this. As I mentioned, there are two important concepts here to remember. The first is this is the exact same format that you are going to have for the exam. So you are going to be presented the images all at once so that you have the opportunity to look at them, go through them in any order that you want and be able to answer them, change your answer. And if you want to go back to them, you do that, and then you will submit it all at once. And you have to pay attention to the clock. You are going to be limited in the time that you're going to have. Like I said, the average for the lab exam is about a minute a question plus 5 to 10 minutes for procedural stuff along those lines. And that is the time for it. So again, you don't have time to lollygag. Yes, this is not for a grade, but I would encourage you to maintain normal Proctorio protocols, have your desk cleared out, don't have any papers around it, don't cheat on it. This, again, the point of this is for you to gauge how you're doing in the class right now, to see how your studying is going and how you're going to be successful. Remember, spelling counts. Remember to be specific. Remember to read the questions carefully. People often lose points, not because they don't know the information, but because they don't read it carefully. Read the questions carefully so that you can answer them correctly. All right. You will need your ID. You will need to scan your area. Again, this is going to be the exact same procedure. The only difference between this test and the real one is the number of questions. This first test during the summer usually has somewhere around 70 questions, I think. So somewhere between 65 and 75 questions is usually a good guess for that. This lab score does not count towards your grade. We will go through it together, but it is 0 points. It doesn't count at all. So again, no pressure, no stakes to this at all. This is just a chance for you to gauge how you're doing in preparing for the class so far after this first week. All right, any questions on the procedures of this? Again, it's the exact same format you use for the Proctorio quiz, the exact same one you use for the chemistry quiz. So you guys should be getting familiar with Proctorio. Any questions on any of that? All right, then I will go ahead and release the exam. The exam is now ready, and let's meet back here, like I said, at 11:15. So I'll go ahead and put this screen back up to remind you. We will meet back here 11:15. So take your break now, or take your exam and then take your break. Either way, be back here at 11:15 so we can discuss the exam. Any questions? All right, I will see you at 11:15.